Cancer is a notoriously heterogeneous, tricky disease. It's very hard to understand at the average level. So the first thing we hope to do with PERSIST-SEQ is answer the question, is it useful to generate a lot of single cell data to understand cancer? And hopefully that answer is yes, and hopefully that leads us to understand cancer, and in this case, persister biology a little bit better. So the central idea being that you don't have to drug a pathway or a gene, but you have to find cell states and understand those before you can properly tackle cancer.
I'm Dylan, I'm CTO at Single Discoveries.
And I'm Mauro, I'm one of the two founders of Single Cell Discoveries.
I am Oriol, I'm a team lead within the R&D department at Single Cell Discoveries.
Single Cell Discoveries was founded about 7 years ago as a spin-off from an academic core facility, so one of the first in the world for single cell sequencing. It was really founded because we found ways to automate single cell sequencing, basically. The Utrecht Institute where we were working started a lot of collaborations, and after a while that became too much for the Institute. They started a facility dedicated to helping people with single cell sequencing, and that's where we had the idea to spin off and create a bigger company that could grow with the field and offer single cell sequencing to more people. That's how we also got into PERSIST-SEQ. We really like the idea of helping out building an atlas of single cell cancer biology. Our main role there is to centralize our processing so that we make very standardized data generated in a very standardized way available to the whole PERSIST-SEQ community and later the whole world. Our main contribution was that we understand very well how to handle samples coming from different sites and of radically varying quality. So PERSIST-SEQ is set up around the idea that it's great to do atlasing, so to generate big single-cell cancer atlases or other organ atlases. But one of the hurdles they all have is that the data is generated in a lot of different sites. So everything flows through SCD, which standardizes the whole procedure. But more importantly than that, we were able to think along how to set up a large sample collection sort of network and how to make it easy, not just for us to process the cells, but also for the sites who collect the cells in a way that they're basically ready to do single cell sequencing without having to put them through a very laborious, complicated protocol.
So initially, when we started the consortium, we had to figure out which was the best way to ship samples over to SCD. We have a lot of different sample types as well. So one solution that was working for one type of sample was maybe not the ideal for other types. We did a lot of piloting. And in the end, we settled for a fixation process, which kind of preserves biology at the collection point. And then the different partners can ship the samples over to SCD, and we process these samples with the same methodology. Basically, we can control a bit more the different batch effects because essentially all the samples are processed in a standardized manner. And so far, yes, we've done over 300 samples already, sequenced almost 2 million cells. So it's looking really good. And we are excited for the coming years where more and more submissions are being prepared. So we deal with all the sequencing in-house for the samples, so we can then see which genes are being expressed in each individual cell.
Eventually, every group will analyze the data in their own way and then feedback the results to the group. But the main things we're using right now to really process and send out the data is first, we map the data using Cell Ranger mostly, because we use xenografts technology. We basically send the data to a Galaxy pipeline maintained by EBI in collaboration with the bioinformaticians from PERSIST-SEQ. And that ensures a standardized pre-processing, QC, and data sharing package. So mostly Seurat is used, which is a very well-known algorithm to handle single cell data. And then the data gets sent to each individual group where those are really biological experts.
SCD recently acquired the machine to perform spatial transcriptomics. We can now profile the transcriptome of a sample while retaining the spatial information of the sample. This is very important in fields like cancer research, where you are very interested in seeing how these different tumoral cells are interacting with each other, but also with the immune system of the tissue. To be able to do spatial transcriptomics, you basically require a microscope first to get the spatial information with staining of the tissue. But then we also need a machine to transfer basically the RNA from that tissue section into a capture slide that we mentioned. And then you need another machine which allows you to transfer basically the RNA from the sample into a glass slide, which will have some spatial barcodes, which allow us to then identify from which position this RNA was coming from. And this is basically part of a 10x genomics kit as well.
At Single Discoveries, we have a number of technologies that we offer. So there are cases where you would have a smaller amount of cells that you want to interrogate. So for that, we have methods such as VASA-seq, which works on much smaller cell numbers and gives you a much wider understanding of genes that are being expressed, including differences in splicing or genetic variants, you cannot do with 10x genomics flex, while PERSIST-SEQ is still compatible with fixed cells. So because of our wide range of technologies, we're generally able to answer whatever question the consortium partners have. We mainly use Illumina sequencing technologies. The main one used for PERSIST-SEQ is a NovaSeq X, largest Illumina sequencer currently out there. So the max capacity is 50 billion reads divided over 2 flow cells. And the main reason why we use this large sequencer because it allows us to keep the costs down, get sort of a high enough throughput and a low enough time to be able to turn over projects fast enough. Consortium partners get that data at a really fast rate, so they're able to design new experiments and keep the consortia and project designs going. It's essential for PERSIST-SEQ to have a large sequencer that's able to process many, many projects.
Atlas building projects like PERSIST-SEQ, where many cells have to be processed and analyzed together, eventually suffer from a very common problem, which is that the data is generated in different sites, in different locations, different protocols. And if you ask any bioinformatician, data analysis is hard enough. If they also have to take into account not just that the data was generated in different locations, but also with different technologies or different variations of one technology, that makes it much harder to really get to the bottom of the biology that we're trying to look at. So it's really important that we centralize all processing so that there's no batch effects, and maybe even more importantly, that we standardize the way the samples are being processed. That's incredibly important later on in data analysis. When you find something interesting in the data, you don't want to wonder, is this a gene that I'm seeing because I use technology A versus technology B? You want to be sure that that's all taken care of, and you can really look at the biology and not the technical artifacts. I think PERSIST-SEQ has a very unique and a very nice combination of academia, industry, and service providers like us, all working together to generate a persister biology data set that the community can use. And the reason we're well positioned is that there's a very wide range of biological questions coming from both industry and academia that get asked from the consortium early on. So we kind of took that into account when building the pipelines, the infrastructure, the protocols. From the ground up, it was made in a way that it would be able to handle a lot of different sample types, organoids, PDX, patient, pre- and post-treatment samples that are very relevant to the biology we're trying to understand. And because we and others in the consortium have a lot of experience in dealing with different sample types, and especially low quality sample types, which is very often the case after you treat something with a drug, that makes us uniquely positioned to do this and do it well. It's not just about sharing the data indeed, which is very important. It's about making the data accessible beyond the end of the consortium timeline, and to explain and make public how the data was generated with which protocols.
So what we're already seeing within PERSIST-SEQ is that we're moving towards more and more single cells being analyzed. The field is moving to larger volume of cells in an easier way at lower cost, which I think is going to be transformative for these type of Atlas collaborations. It's going to be much easier to understand complex processes because you're able to just generate so much more data in a much shorter amount of time. That being said, I think single sequencing has one role to play in it, but I think spatial transcriptomics is sort of the other factor that will be complementing the data generation.
So next steps are, one, keeping the lights on, so making sure everything functions properly and data keeps being analyzed in a rapid pace so we can iterate on the next round of experiments. But it's just also about thinking ahead. We have a goal of at least 5 million cells. And while we could reach that easily by just generating a lot of data from one sample type, the next steps are really identifying where the gaps are, so what cancer types we are missing in the consortium, making sure we get samples from those. So either stimulating the labs that could produce those samples, thinking along with them what type of samples to produce, or finding other collaboration partners that could help us fill those gaps in the data we're trying to produce. And then the last step is what we always do at SDs, is keep being on the cutting edge of single cell and spatial technology, so that if there's a question that can be answered with one of the current technologies we have, we add a new one. So we can keep moving forward and keep adding interesting sample types that can help us understand persister biology. I think the main immediate impact we'll have is that we have a very clean, nice, high quality data set with a lot of different samples and sample types. that people can analyze. This is a data set that's too big to be analyzed even by a consortium like PERSIST-SEQ, which has many groups. Is really something that a lot of people from different angles should look at. So the first impact is giving that or producing that data set for basically the whole world to analyze. Secondly, it helps people understand that cancer biology is maybe not just about a gene or a receptor or a pathway that if you find it, it's a silver bullet and you fix the problem. Sometimes that's true, but sometimes it's not. So it's helping people shift from that mindset to the mindset of maybe there are cell states that are temporary, that just exist in time and then they go away again. So that we can try and understand cancer in a different way and treat it in a different way. So as we zoom in on single cell data, the question, what is a cell type, becomes very philosophical almost. Is it a cell type? Is it a cell state? Is it a blip in time that's not really significant? Or is it something that's really there for long periods of time and prevents cancer cells being killed by therapy, for example? Beyond having the data set for people to analyze and finding interesting targets or cell types is helping people think about cell states and as something that's druggable.